INO-5401 is a DNA vaccine, actually, and it's a novel way to introduce an antigen and create immunogenicity against an antigen by injecting a piece of DNA or a plasmid into a person, and they do that with this particular drug intramuscularly. And ultimately, what that DNA plasmid does is it gets taken up into the host cells, the host muscle cells, and that the plasmid then turns on transcription and production of the specific protein that it's encoding. So the INO-5401 specifically carries DNA plasmids that are predicted to be tumor antigens. So for glioblastoma, it carries DNA plasmids for human telomerase, WT1, and PSMA. And these are three proteins that are highly overexpressed in most glioblastomas. And the principle is that it is a tumor vaccine and will stimulate the antigen response to these tumor-associated peptides. And it does that in a novel way by injecting the actual plasmid DNA rather than injecting peptides, which is a classic way of creating a tumor vaccine. And so the mechanism by which they introduce the plasmid is novel, and I think the principle of injecting DNA into a person is novel because the way that the antigen is produced, instead of just the peptides themselves, the DNA is inserted, and the host can actually continually, potentially, produce that antigen. And not only that, it's what they will do as part of this trial is repeated injections of that DNA vaccine. And so, to me, one of the interesting things about that particular DNA vaccine is the mix of peptides, the mix of tumor antigens that they're using in this product. So WT1 and PSMA are proteins that have been studied in previous studies to be overexpressed in glioblastoma. So these are tumor-associated peptides that we think a majority of glioblastomas will overexpress. But one of the things that I think is particularly interesting is that it's going to express human telomerase. And glioblastoma have very recently been characterized on a genetic level through multi-region sequencing and paired tumor sequencing. And in principle, what we think is important in terms of finding a target, something either whether you're going to drug a target with a target inhibitor or with an immunotherapy agent, is that you want that target to be in every single tumor cell within the entire tumor mass and also be retained. If the tumor grows through primary therapy or recurs, that target to be retained. And in glioblastoma, I define glioblastomas very differently. I think there's not just one type of glioblastoma. There's several different subtypes based on the molecular makeup, so the genetic driver mutations in these tumors. But if you take all of glioblastoma, there are glioblastomas, a very small percentage have driver mutations in IDH, and a very small percentage of glioblastomas have driver mutations in BRAF. If you exclude those, the vast majority of glioblastomas have driver mutations in the promoter region of TERT, or the human telomerase gene. And it's been shown that that TERT mutation is clonal throughout the tumor, meaning it's in every tumor cell, and also it's retained at recurrence. So it is a true clonal driver mutation, and therefore it really represents an incredibly attractive target for really any modality of therapy, whether it's immunotherapy or targeted therapy. The other interesting thing about human telomerase is that it has been studied as a vaccine for quite a while. And so in principle, it's an immunogenic antigen, and it's a clonal driver in glioblastoma. So therefore, it really makes targeting human telomerase an attractive strategy. So that's one of the things that I think that make that particular vaccine an interesting strategy in glioblastoma.